and my topic of presentation for today is cell cycle and cancer. So, what we all basically know about cancer is uncontrolled cell cycle or uncontrolled cell division. So, my topic will generally cover the cell cycle in detail and the scope how cancer derives its formation in a cell cycle. Actually, scope is not a good word, we should say that mishappens that may lead to cancer. So, cancer is compared by some scientists as a vehicle whose fuel is accelerated, always on, and brakes are failed. The accelerated fuel is compared with the protoncogene's activation, which leads to oncogene, that is gain of function, and then brake failure is compared with loss of function of the tumor suppressor genes. We will see those in detail. Although I will not consider the oncogenetics and tumor suppressor genetics, but I will try to compare them how these cytosolic functions or cell cycle functions lead to these gene activities. Moreover, this circular table is representation of how a cancer cell is different from a normal cell. Okay, so cell cycle. Cell cycle is composed of all these phases. A freshly derived cell from a mitotic cell division can enter G0 phase, which is basically cell arrest. The cell's growth is ceased, or it can come into G1 phase, which is the first phase of the cell. G1 phase and G0 phase can do vice versa because certain cytosolic factors are involved. Cell signaling is involved for the growth. Now, in G1 phase or first growth phase, the cell grows in size. It produces mRNA delta required for the S phase DNA replication, and organelles also form better. Then it further reaches S phase, and in S phase the genome is replicated in a highly conserved and regulated manner. Thereafter, the G2 phase, the cell grows furthermore in size for division of mitosis, and certain proteins that are required for mitosis are organized. If we consider the M phase or mitotic phase, since we are not considering the somatic cells, so I will focus on mitosis instead of meiosis. Meiosis might have different cellular modification for cancer, but let's focus on mitosis for this case. Mitosis begins with prophase, where the chromatin condense into chromosome. They are visible. Centromere movement is observed, spindle fiber formation is observed. Thereafter, in the late formation of prophase, the nuclear envelope breaks down, and for metaphase, the chromosomes are aligned on the middle of the dividing cell, that is known as metaphase state, and spindle fiber are ensured at the end of the metaphase before we proceed into anaphase. Then at anaphase, the chromosomes are pulled apart into two poles of dividing cell. Thereafter, the telophase is activated. The nuclear envelope begins to reappear, and the actin and myosin protein rings start to contract to divide the cell, and further after cytokinesis, the new cells are formed. Now, for this cycle is repeated again. I have picked 4 places. The 4 places are representation of checkpoints. Since we majorly study 3 checkpoints, I have included one checkpoint that is between S phase to G2 phase. It is not majorly studied, but what happened is certain ATR proteins they inhibit the FOXN1 gene. It is a mitotic gene, and it is kept inhibited until the whole genome is replicated. But its activity is not studied further, so I will exclude this, and I will talk about the basic G1, G2, and M checkpoints. For the beginning of G1 phase to S phase checkpoint, at the end of the G1 phase, what happens is that our 2 transcription factors, DP1 and E2F, they are kept suppressed by histone acetalysis and RB2. So, with impact of cellular modifications and signals, 2 cyclin-dependent kinases and cyclases bind together. They phosphorylate the RB protein, which in turn releases its attachment from the transcription factors, and the transcription factors can further occur into S phase. Right? So, if you consider that portion covered, which is highlighted in the corner, I want you two complexes produce the PLK1. To produce phosphates, there is CDC25. CDC25 in turn activates the CDC2, and thereafter this complex is emptied into mitochondria. Now again, I'd like you to remember that this is summarized, and this checkpoint is furthermore affected by certain other factors. DNA damage is kept in check. DNA damage can basically produce two major responses: the DNA PK response and the ATM-ATR pathway response. What happens is when ATM-ATR pathway response is activated, it leads to formation of certain CHDs that are kinases, and those kinases inhibit the CDC25. And when CDC25 is inhibited, the CDC2 will already be inhibited, because that are the phosphates which helps them activate. And the other is DNA PK pathway. DNA PK pathway lead to formation of P53. P53 holds the cell to lead into mitosis. And if DNA damage is repaired, then the cell can further lead into mitosis, otherwise it will not. For cancer cells, the first primary oncogenes, MD1 and MDM4 oncoproteins, they inhibit the function of P53, and thus the cell cycle progresses into early mitosis. Oh, one more. I'd like to focus on the written part, caffeine here on the ATM-ATR pathway. In vitro, when it is observed that caffeine and more genotoxic substances affect the ATM-ATR pathway, which in turn can lead to certain therapeutic processes, but when I read into it, it does not affect in vivo. So drinking caffeine will not affect the ATM-ATR pathway that's working on at our bodies. Next is spindle assembly checkpoint. Spindle assembly checkpoint is activated by the open kinetopo. Open kinetopo invites certain spindle assembly proteins to bind into it, which activates the mitotic checkpoint complex, which is those four proteins where CDC is kept inactivated. When spindle assembly checkpoint allows the kinetopo to bind with the microtubules, the ends of the microtubules, that is alpha and beta, form a cascade of protein reactions that lead in dismantling of the mitotic checkpoint complex. When this complex is deactivated, CDC activates, and it leads to formation of anaphase promoting complex. When anaphase promoting complex is activated, securin protein is degraded to form a protease called separase. Separase allows the cleaving of the chromosomes into two mitotic end. Thereafter, degradation of cyclin B leaves our CDK1 inactive, and this allows the further mitotic exit. One thing I forgot to tell is that our cell cycle is regulated by the concentration of cyclins and its association with CDKs. CDKs are always present in the cell, but cyclin concentration may vary, and that is the deciding factor how everything proceeds. Also, in cancer cells, CDC20 activity is observed in high activity. It's overexpressed, which works as oncoprotein. But it is also target that if we inhibit the activated CDC20, then it can lead to some therapeutic changes, which are not cytotoxic for other cells except for cancers. So these are the observations which I will conclude in words. Cyclin and CDK's expression decide whether the cell will become cancerous or not. Okay. P53 expression will decide whether the cell growth is inhibited or not, or the gain is impaired or not. And here is the detailed data that has been collected from a review paper, where what genes of what cancer types does what type of gene alteration to lead what type of CDKs and cyclin mutations. And it can be looked further into if you want to evaluate that. That's